Hello, everyone. We're going to get started. I'm Kathy Antela, Vice President of Education at the Immunodeficiency Foundation. And before we go any further, I would like to introduce our Executive Vice President of Programs and Services, Hector Garcia. Hector.
Thank you, Kathy, and good evening, everybody. And we usually write a script for every forum that we have, and we go. Takes a lot of people to plan for it, and this is totally unscripted. So I was not supposed to be here. We kind of take turns in terms of who does a welcome, but it hit me that it's Tuesday before Thanksgiving, and here we have over 100 people supporting IDF by your participation of our forums. We have a physician that it's fully dedicated to support us tonight. We have staff. Even though we might think that when we met in person, it was a little easier than virtual. It actually takes more people to plan a virtual forum than it did to plan a meeting in person. So there is more staff behind every forum that you see and that you participate in, and a lot of them are here tonight to support this. So it just kind of hit me, and I'm not going to get emotional or anything, but it's a Tuesday before Thanksgiving. A lot of people would be in airplanes, would be traveling, to see their families. Tomorrow would be like the highest traffic day in the year, and here we are on a Tuesday night, both of us, you and our staff and our physician, getting together to try to improve quality of life. And it just means so much to us, and I mean to IDF, that we have your presence here tonight, that you've been supportive throughout the year, and that we're doing everything we can to keep you safe, and that's our priority. I also wanted to say thank you, as we're in the a little bit of a gratitude mode, to our sponsors for making this possible. They're here with us tonight also. So I just wanted to butt in. I'm not even dressed up. My dog Lola didn't know there was a plan for me to show up tonight, so she won't come down for treats. But I just wanted to thank you, our audience, our community, for being so supportive. It's been quite a difficult year for all of us, and to be able to have your support, it's amazing. It's amazing, and you let us into your homes. You get to you help us get to know you better, and here it is 2 days before a time where we wouldn't even think to do anything like this, the Tuesday before Thanksgiving, because nobody would show up, and here you are, and we're getting close to we'll get 150 people. I'm thinking, and this is amazing. So I just wanted to say thank you, our gratitude to you, our community, and I also need to say the incredible dedication of IDF staff, and the physicians that support our program. It's just amazing. It's awesome. There's folks here that, could have taken the week off, could have taken today off, and they're all working tonight, to provide you with, what will be an incredible program. And we couldn't do this if you were not here. 7, 8 people, 7, 8, or 9 staff of us doing this between ourselves wouldn't work. So it's all because of you, also because our sponsors that we're here. So my heartfelt thanks, to you, our community, and please stay safe, continue to everything, you can to keep yourselves healthy. And hopefully we can continue to bring you closer to us through our programming and help you with, whatever you need to be able to, continue to be safe and sane in such difficult times. So that's all I have. I probably took a little longer than I should, but I was just walking around the house and I looked at the numbers for tonight, and I couldn't stop myself from interrupting Kathy and saying thank you to our audience, our physicians, our industry partners, and our staff. And they're all here tonight with smiles on their faces. They're excited to serve you, and that's our biggest asset. So anyway, thank you. Don't leave. I'm going to say good night. Don't go anywhere. And I'm sure you will enjoy tonight's program. Kathy, thank you for letting me interrupt you. You go ahead and take charge along with everybody else. And please, stay safe, all right? And hang in there, all right? Good night.
Thank you, Hector. Enjoy the upcoming holiday weekend. Get some R&R. Take care. Next, a quick disclaimer. The information presented during this event is not medical advice, nor is it intended to be a substitute for medical advice, diagnosis, or treatment. Always seek the advice of a physician or other qualified health provider with your questions concerning a medical condition. So on behalf of the Immunodeficiency Foundation, welcome to our 42nd virtual forum, 42 of them. Tonight's presentation is overview of genetics and primary immunodeficiency. To our return attendees, it is great to see you again. And for our new participants, we are so glad you're here and do hope that you will join us again. Our forums are designed for you and by you. They're designed based on the feedback that you provide by completing the evaluation you're going to receive via an email after this event, and also through your support and participation by being with us this evening. So thank you. Now, as you can see from our mission, IDF is dedicated to fostering a community empowered by education. We want you to remember that IDF is committed to our community, serving you as a trusted resource through the use of technology and innovation, that we are here to give you the tools and information you need to become empowered. And at IDF, we value integrity and inclusion and offer you our compassion, understanding, and support to emphasize that you're not alone on your journey. We are with you, we're together every single step of the way. We are so fortunate to offer these forums and additional events to our community due to the support of our sponsors. And it is because of their partnerships and contributions that IDF is able to offer programs and services so that everybody is better informed. This includes Core Service Leaders, CSL Behring, Grifols, Takeda, Core Services Supporter, Octapharma, Core Service Sustainer, Horizon Therapeutics, Core Services Contributor, Leadient Biosciences, and ADMA Biologics, National Sustainer, Accredo, National Patrons, Diplomat Specialty Infusion, Kedrion Biopharma, CVS Specialty, Quorum CVS Specialty Infusion Services, Carew Medical Systems, Caba Fusion, and Celio Health. Many of our sponsors do have plasma donation centers, and we encourage you to please visit their website for more information about how you can make a difference during this period of time. Now, at this time, I would like to invite Ellen DiGirolamo from CSL Behring to say a few words. Ellen, you want to unmute yourself?
Okay, thanks, Kathy. Can everybody hear me okay?
Yes.
Okay, thank you. Hi, everybody. My name is Ellen DiGirolamo, and I'm one of the area managers at CSL Behring, and I represent our immunoglobulin products, both Hizentra and Privigen, and I am very excited to be here tonight. I want to thank the IDF for continuing to host these important online forums and for adjusting to this virtual environment so that the community can continue to learn from each other and support one another during this challenging time. Of course, I look forward to the day where we can all meet again in person safely. But for now, I'm just very happy to be here tonight to support patients in any way possible, either by offering information on our resources or answering any questions at all that you have in the breakout rooms this evening. So thank you so much for having me, and I'm looking forward to the speaker tonight.
Thank you, Ellen. Next, I would like to invite Victoria Martucci from Grifols for a quick word from our sponsor.
Hi, everyone. My name is Victoria Martucci. I am with Grifols with Xembify, our 20% sub-Q, as well as Gamunix C, which is our 10% product. I just wanted to thank the IDF. It's an honor and a privilege to be here tonight. I am joined by Debbie Manning, who is one of our nurse educators tonight, and we will both be in the Grifols breakout rooms later to answer any questions about patient resources that we have, patient assistance programs, support programs. Just wanted to thank the IDF again for everything that they've been able to do with all these virtual events during the pandemic. And it is an honor to be here, so thank you so much.
Thank you, Victoria. We look forward to hearing more from you and all of our sponsors during the virtual exhibit hall today. Now, this evening's topic, overview of genetics and primary immunodeficiency, is presented by Dr. Lori Broderick. Dr. Broderick is a board-certified allergist immunologist at Rady Children's Hospital in San Diego and is an assistant professor of pediatrics at the University of California, San Diego, with special interests in primary immunodeficiencies and autoinflammatory syndromes. So please join me in welcoming Dr. Lori Broderick.
All right. Thank you for the introduction. We are doing things a little bit differently because I'm sharing my own slides this time. I'm thrilled to be back at an IDF forum. This topic I've presented in the San Diego forum, I think last year and perhaps the year before. And it's something that I think is just really important for everybody to understand in terms of benefits, limitations, and really what genetics can mean for primary immunodeficiency. I do have a couple of disclosures. I do work at UCSD and I have a research lab where I study rare immune diseases in both the immunodeficiency and autoinflammatory realms. And so I have research support, but I will not be talking about any of those projects or the participation in advisory boards during the course of this presentation. So I'd really like to do tonight is give you an overview of what genes are, what it means in the immune system, starting it from the very basics of what is DNA, what is a gene, how does that become a protein and turn into a phenotype, how do genetics and primary immunodeficiencies relate to one another, and what are the benefits and limitations of testing, and how could you potentially harness the power of genetics for understanding your own disease. So just to give a brief overview, if we think about the immune system and how it changes throughout our life, we begin as little babies. We actually require our mother's immunoglobulin to get us through those first few stages as our immune system starts developing. It changes throughout the first years of life as our B and T cells continue to develop. It changes with certain life events. If you're a woman who becomes pregnant, you're going to have certain immunologic changes that are necessary to support another life. And then as we enter the more seasoned years of our life, our immune system changes further. And despite all of those changes that are constantly happening in our bodies and each of our cells, our genes remain largely the same. But genes determine how we respond to the rest of the world around us. They tell us what we're going to look like. how we taste foods, what foods we like, what foods we may have an allergy to, if we develop pollen allergy or other environmental allergens. And then finally, how we respond to the microbial community around us, both in terms of maintaining a symbiotic relationship with the bacteria in our gut and how we approach infection. And immunodeficiencies, our understanding of the immune system and our understanding of genetics actually have a long history of partnering together. So I want to go way back to the 1950s when Colonel Bruton first reported the first known immunodeficiency and gave it a name. He saw a little boy about 8 years old who was found to have extreme susceptibility to upper respiratory infections. He was able to do some experiments very similar to those that I do in the lab every day, and showed that there was a lack of immunoglobulin in the patient's serum. He didn't know that it was IgG at the time, but this was the first sign that there was something different about this patient. And he was actually the first to give somebody immunoglobulin replacement to show that not only could he replace that missing protein, but it could help control the life-threatening infections. So this is the first description of a primary immunodeficiency, the first known instance of using immunoglobulin replacement. And all of this was done without actually knowing what was causing the disease. And it wasn't until the early 1990s, in 1993, when 2 different groups independently published finding the genetic basis for Bruton's Agammaglobulinemia, now called X-linked Agammaglobulinemia. And that gene was BTK. And what they found is that a specific genetic mutation in BTK stopped the progression of B cells from the precursor cells through the pro-B cells and prevented it from going on to the pre-B cell stage, which meant that the cells weren't present to be able to make the immunoglobulin. So that first finding that Bruton had 40 years prior, they finally found the answer to. And the reason this is important is because the immunologic field is still fairly young, and we are learning new things about the immune system and about genetics every single day. And that doesn't mean that we can't still approach treatments and approach improvements in quality of life in the absence of a genetic diagnosis. So let's start at the very beginning. What's a gene? Well, to know what a gene is, we have to understand what DNA is. So DNA stands for deoxyribonucleic acid. This is a complex molecule that contains all of the data necessary to build and maintain an organism throughout their entire life. Now, in a multicellular organism, like a human or a dog or a cat, almost every cell in the body has the full set of DNA required to make that organism and to keep them alive. But DNA is really the primary unit of heredity, the things that help us transfer information from one generation to the next. However, it also leaves a little bit of wiggle room for, dare I say, error, but also opportunity, so that slight genetic changes can happen from one generation to the next to tweak things and possibly make things better, but also sometimes knowing that mistakes can happen. So DNA is stored as a code of just 4 chemical bases. So adenine, guanine, cytosine, and thymine, the famous AGCT code that all of life is built upon. And the organization of these 4 bases is most stable as the famous double helix, which is shown on the right side of the screen. If we took all of our DNA from a single cell and just aligned it one after another line to line, it would reach almost 2 meters, so almost 6 feet. That means that it has to be tightly packaged within the cell. So the DNA base pairs are wrapped into a helix. Those are wrapped around proteins called histones that then form nucleosomes and are tightly wound together to form the chromosomes, which are inside the nucleus of the cell. The genes that are made of DNA are technically the functional unit of inheritance, the things that we can track from one generation to the next. But a gene is actually split into a few components. The exons, which is the coding region, or the part that transfers the information, and the introns, which is the non-coding portion. And for a long time, we thought that the introns were just garbage. It was cut out when we were looking at the DNA. And now what we really understand is all of that intronic region actually can have important regulatory sections that determine how and when a gene is expressed, in what cell type it might be expressed in. And all of that information tells us more about how cells function and how we function as people. So to get from DNA to a human, we have to go through a number of different steps. So we've got DNA on the left side here. It then gets transcribed to RNA. So those introns, the non-coding parts, are cut out. The exons are fused together. And then those are translated into a protein. Proteins from multiple genes then interact with one another to create a phenotype, or the picture you see. So in this particular example, you see a gray bunny. If you have a variation in any part of that gene or a mutation, you can get changes in the phenotype. So if you have this little red star here as an example of the variant, rather than getting maybe a gray bunny, you get a bunny that has all sorts of different colors, some that are perhaps a little bit smaller and look a little bit more like guinea pigs for some reason, but it changes how the proteins interact with one another and thereby changes the outcome. So you can think of these variations or mutations as almost spelling errors. So I'm out in San Diego, and so of course we love surfing in the ocean out here. So the example I'm going to use is, instead of ACTG, we're going to use surfing as the protein that comes together. If you have a substitution, meaning that one letter changes for another, you can get different words. Instead of surfing, you get surfing with an E, or surging. These are both new words, but they don't have the same meaning as the original word. An insertion can be introducing one letter or a few letters, changing again the meaning of the word and whether it's even sensical. A deletion could change by deleting just one small part and creating, again, a word that doesn't exist, or deleting a few letters and creating a new word, again, with a different meaning. So instead of starting with surfing, we end up with sing. And then you could have a truncation where you lose the end part of the gene and get an early stop, and you get a much shorter portion. These genetic variants can happen anywhere in the gene, and then they all affect the outcome. Other genetic changes, besides these individual base pair or a few base pairs, can have more serious effects. You can have big deletions where you cut off entire exons, either from the beginning, the end, or the middle of the genes. You can have copy number variations where instead of having one copy of a gene, maybe you have 2 or 3, so you're producing extra copies of a protein. Or you could get deletions of entire genes. So rather than having one inherited from each parent, you only have one, either from your mom or your dad. And finally, you can have large deletions or duplications to the extent of an entire chromosome. And so this is called trisomy if you have an extra chromosome, or monosomy if you have only one copy of that chromosome. And so probably the example most people are familiar with is trisomy 21 or Down syndrome. And so obviously having extra copies or too few copies can have an impact on the individual. So how do we inherit these genetic changes? Well, we know that we have to have an egg and a sperm that both contain genetic copies coming from the parents. And there's a number of different ways that things can be genetically inherited. So in these particular examples, what we're calling the affected gene, the one that we're following, is in purple. And so in this case, when you have an autosomal dominant genetic change, we have the genetic change here on the chromosome, it's purple. And this father can pass that change to 50% of his offspring. And it doesn't matter if it's a boy or a girl, he will pass that gene 50% to them. They will get their other copy from their unaffected mother. And so these two kids, potential kids, would have the exact same phenotype as the father then. In an autosomal recessive disease, the parents are carriers for that genetic change. So meaning they have a genetic change on the same gene, doesn't have to be the exact same change, but just on the same gene, and they're unaffected. But if they both pass that genetic change to their offspring, which happens 25% of the time, then you get an affected child because they have 2 copies of the mutated gene. 50% of the kids will look just like the parents and be carriers, and then 25% will have no carrier phenotype whatsoever. If that genetic change happens on one of the sex chromosomes, so on the X chromosome, for example, that changes how the genetic inheritance pattern looks. So in this particular example, we've got an X-linked recessive trait where the mother is the carrier. So we see it here on her X chromosome. So the difference between, a biological male and female are that you have in the male one single X chromosome and one Y chromosome, and then a female has 2 X chromosomes. So if she passes that X chromosome to her son, he becomes affected because he needs to get his Y chromosome from the father. If she passes it to her daughter, then the daughter is a carrier. And then there's 50% of the kids who wouldn't be affected. If this boy then goes on to have his own family, he has his affected chromosome here, which then he can pass on only to his daughters because he only has one X chromosome to pass on. They will both be carriers, like the mother in the first example, but he can't pass this on to any sons because he needs to pass them the Y chromosome. These sorts of changes and this inheritance happens in every single person. That's how we've got all of our genes. Sometimes it leads to disease. And so how can we use this information in primary immunodeficiencies? How does knowing these genetic changes help us? Well, in some cases, it can help confirm a diagnosis. It can help define prognosis. It may have implications for treatment, and certainly may have implications for family planning in the future and how we think about our current families. So if we talk about genes and diagnosis first, we know that there are more than 400 different disorders that have been identified affecting the immune system. This is from the practice parameters from 2015. All of the diseases that are listed on this page are known to be diseases of the immune system. Not all of these diseases have known genes associated with them. So again, we're still learning more every day. Some diseases have only one known gene. Other immunodeficiencies have multiple genes which could cause disease. And then, as I said, genes are being identified every single day. So just to give you some examples, if we think back to Bruton's Agammaglobulinemia, that's an example where one genetic defect causes one disease. You have to have a mutation in BTK to be diagnosed with Bruton's Agammaglobulinemia. Can you have other causes of Agammaglobulinemia? Absolutely, but it wouldn't be called Bruton's because it doesn't have this mutation. In contrast, common variable immunodeficiency, or CVID, has a monogenic or one gene cause where things are inherited in about 2 to 10% of families. However, many different mutations have been linked to that monogenic cause. So we've got a couple dozen mutations here in all different genes, all which affect different aspects of B cell development that can cause a CVID picture. So what does this mean in terms of prognosis and treatment? Well, as I mentioned before, you don't have to know the genetic defect to necessarily get treatment, but it can help you make decisions. So some genetic variants affect prognosis because certain types of mutations may be more severe. Some carriers may have symptoms. And so one of the examples that we use most frequently in the clinic is thinking about CGD, or chronic granulomatous disease. In this particular case, we have a schematic of what the protein looks like. You've got some parts inside the cell, some parts outside the cell, some parts in the cell membrane. And depending on where the mutation is, it affects how much residual enzyme function you have. So some of these mutations only affect enzyme function a little bit, so people aren't as symptomatic. And other mutations knock out enzyme function completely, which give a full-blown picture of CGD. The type of mutation and the exact variant can also have implications for the type of therapy. And this is specifically important when we're thinking about hematopoietic stem cell transplant. So in SCID, or severe combined immunodeficiency, we have learned a lot about genetics since the onset of the newborn screen because people are being diagnosed earlier. If you have this type of SCID, Artemis SCID, called spy mutation in DCL-RE1C, maybe you want to enroll in a gene therapy trial. However, if you have ADA SCID, we know that patients have been treated not only with stem cell transplants, but also with enzyme replacement or with gene therapy. And so it changes how you may think of your different treatment options if you know what specific mutation you have, if it's been described before. Because of all of these variables, genetic counseling is important at every stage of considering whether you want to get a genetic test, both from before the test is ordered to when the test is in process, to when the results come out. And that's important not just for you, but for your family members who may be involved in the process as well. So what's the implications for gene and family planning? Well, it actually starts with the family history when you go to your doctor's office and they say, does anything run in your family? Does anybody in your family have a history of immune deficiency? Because that gives us information about what type of immune deficiency you might have and how the gene may have been inherited. But it's not the whole story. A family history may reveal the inheritance pattern. So if you and your father, for example, had a similar disease, we could say maybe you inherited it from him. But disease penetrance varies. So depending on the type of mutation you have, the amount of residual function there is, people may not be affected to the same degree of severity. You can also have what's called a de novo mutation, meaning that you are the only person in your family to have that genetic change because it happened to you for the first time ever, and so nobody else would be affected. We can also have what's called variable expressivity, meaning that if certain genes are only expressed in certain types of cells, that people may have different levels of severity of their symptoms at different stages in their life. To complicate the picture even further, we now know that something called somatic mosaicism exists. So on the left hand is the inheritance that we usually think of. We have a gene, a genetic change that's in the mom or the dad. It comes through either the sperm or the egg. So the zygote and all of the cells have all of the same genetic information. As those divide into an embryo and then ultimately into a fetus and an infant and then an adult, every cell in the body has all of those changes. But we've known from the cancer world that genetic changes can happen throughout our life. We can get a spontaneous mutation in one of our cells that then creates a tumor, meaning that tumor is genetically different from the other cells in our body, and that's shown here. So we had somebody who was growing normally, got a spontaneous mutation, and maybe developed a tumor. What we now learned in the last just couple of years is that sometimes a mutation happens in the zygotic or the embryonic stage, which means that only a portion of the cells are affected. Depending on where that happens, it may mean that only some of your tissues are affected. So perhaps, the mutation happens so that the thyroid was affected, but not the blood cells. And that has implications for trying to find those mutations within a person. This was actually first described in an immune dysregulatory disorder known as the autoinflammatory diseases, where patients would have what seemed to be full-blown autoinflammatory disease, and you couldn't find a mutation. And it was because only 4% of their cells were actually carrying that mutation, but it was enough to cause systemic disease. And this creates a lot of challenges because not only do you not have the family history behind you to support the inheritance pattern that you're seeing, but you may have disease developing later in life and technically difficult to diagnose disease. So now we know how genes work. We know how genes are inherited. If we have a certain phenotype, what kind of genetic test should we get? What can we learn about our own genes by the tests that are available? So there's a number of different genetic tests, and the decision should really be based on the clinical history, other testing results, and what would be the most informative for your particular situation. So certain genetic tests look at the chromosome level. Others look at single genes or gene panels. Those are the ones that are more commonly used in the primary immunodeficiency world. And then there's exome and genome sequencing, which look at more information from your entire genetic makeup. And so to break this down a little bit, to explain the differences between the targeted sequencing, which would be single gene or panels, versus whole exome and whole genome sequencing, I think this picture really nicely shows the amount of information you can get. So if you have targeted sequencing, you've picked one or a few genes, and you just look at that 100 times and say, can I find a genetic change in those specific areas? If you look at the exome, you're looking at not just a couple of genes, you're looking at all of those exons for all of the genes across your entire genetic makeup. If you look at the genome, you're looking at both the exon and intron areas. And so in the table below, I have just some of the differences between the panels and what their benefit can be. So the panels in single gene sequencing have the best sensitivity because they're looking at every single area of those very specific parts multiple times. So it gives you more reassurance that when the sequencer goes through and finds a genetic change, that you can believe that genetic change. They can look for changes in 1 up to a few hundred genes. The exome will cover all of the genes, so between 4,000 and 20,000, but it doesn't look at any specific gene as highly as a targeted sequence does. In some cases, if you have an unusual presentation or you can't figure out what gene might be causing your symptoms because it's affecting multiple different organs, sometimes an exome can help figure that out. Exomes and genomes tend to be used more in research when you're trying to identify a new gene that's never been described with disease before. And then in genome sequencing, not only are you looking at those 20,000 genes, but you're also looking at all the non-coding regions that we're still trying to learn about. So it's often used in research situations to try to see, okay, is there a change in this intron? Could it be affecting why the gene turns on and off? Could it be affecting gene regulation? Those are much more difficult to prove and to link because the introns have a lot more variability between people. Importantly, all of these have about a 20% success rate, meaning that if you get a genetic test, there's a 20% chance that you will get an answer that makes sense. And I like to be very upfront about that because I think it's important to remember that we still are learning things about genetics every single day. So to summarize that sort of complex figure, I really love this example because you can think of getting an exome or a genome, excuse me, as watching a football game from beginning to end. An exome is like reading about the game highlights the next day, and a targeted panel is more like watching the Twitter feed go by and getting those short blurbs. So what are the challenges to using a genetic test and even getting it? So cost, of course, we'll talk about what the costs are. Accessibility, so who can get one and who can't. And then how do you interpret those results? So the price of genetic testing has actually come down significantly just over the last few years. So we thought that, based on Moore's law and the ability of processing speeds to increase over the last couple of decades, that would directly translate to genetic testing, except genetic testing has accelerated in terms of our ability to make the reagents more cheaply and to process the data. But in spite of these lower costs, if you've tried to get genetic sequencing or you know somebody who's been sequenced, it can still be very difficult. And so part of that is because the real costs of sequencing are not just in the computer reading sequence. It's not just in the reagents that you need to isolate DNA and put it onto the machine. There's a lot of time involved with clinicians, genetic counselors, talking about all of the different steps of the process. There's the bioinformatics processing, which is still very much human-based to go in and say, yes, this looks like a real genetic change, this does not, because we want to be careful about not giving out false information that could affect somebody. We need to do confirmatory testing. So even if you look at that specific read or that specific area of the gene 100 times, you want to be pretty sure before you tell somebody they have a genetic mutation that could affect their treatment or their prognosis. You want to be sure that change is real. And you do that by another level of genetic testing called Sanger sequencing. That's considered the gold standard. Finally, for some people, if they get a bunch of different genetic changes and it's hard to figure out whether it's linked to their disease, they may go on to get confirmatory tests. They may need endoscopy, imaging or surgeries trying to figure out, does that genetic change prove why they have the disease they have? And that's another financial burden for that patient. And then finally, we have to think about where are we going to store all of this data? Our cells have packed things really neatly into chromosomes to store into their nuclei, but we need to store things securely in the cloud. And there's certainly nothing more important than our own cybersecurity right now. Beyond cost, we have to think about accessibility. So insurance can be a barrier for a lot of people. Depending on the type of your insurance, they may call genetic testing research, and they don't think of it as a validated test because there are still so many unknowns. That means that in some cases they may deny it completely or they may only partially cover the cost. So depending on what your home finances are, that may mean that some people are able to swallow those out-of-pocket costs, whereas other people are not as able to. Because of this, a number of commercial genetic labs have been offering lower costs to partner with the families to try to provide more sequencing opportunities to people more evenly, regardless of income. And that becomes important because the more people that are sequenced, the more information we know about what could be disease causing and what may not be disease causing. And then we have to think about the timing of results. The amount of time it's taken to get sequence back has shortened as well, but we need to think about how that will impact your treatment and how soon you need that treatment. So let's say we get through all of those barriers, you've been sequenced, you get a report, your physician gets a report they want to discuss with you. What does it mean? The biggest challenge to any genetic test is the interpretation, because it requires the correlation between a positive finding and the clinical disease picture. And it becomes difficult because all of us in all of our genes have genetic changes that make us who we are. So if we look across the board at our genome, we all have about 4 million changes that make us different from what's called the reference genome, or the person who was initially sequenced. So the initial was J. Craig Venter. And so if we use him as the reference sequence, obviously I am going to have many different changes from him because he is a white man and I'm a woman. Beyond those basic changes, we then look to see how many of those changes could be in coding regions in the areas that we know will affect protein function. Well, that could be about 20,000. How many of those change the meaning of the protein? So going from surfing to sing, say, those are another 10,000. And then how many of those could be just novel changes that have never been reported before? That's still 2,000. And so to compile all of that information, what we've had to do is take people who've been sequenced, take their mutations, put them into cell lines, put them into mouse models or other animal models, use human primary cells to figure out, if I make this cell change, do we get the phenotype that I see in my patient? And that's what takes a long part of the process. And that's very important in terms of the interpretation of, does a genetic change actually link to disease? So it's not as simple as just sequencing. It's a matter of proving that the circuit comes together. And so despite that, if somebody gets a genetic report, it may still say, my test was negative. And you say, well, Dr. Broderick, you just told me that there are 4 million changes that make me different. So why did I get a negative report? And the reason that you get a negative report are because there are different types of genetic variations. And for various legal issues, for various correlation issues, those things determine how reports are released by different companies. So first, we've got findings relevant to the reason that a genetic test was performed in the first place. So if you have a genome that was performed and you get a hit on a disease gene that makes sense, that is related to a known pathway, or it's been described in another family, then you're going to get what's called a variant listed. If you have what are called incidental findings, meaning we are trying to investigate why you have a primary immunodeficiency, but we found out that you're a carrier for a gene that puts you at a higher risk for Alzheimer's. Do you get to know that? Should you get to know that? So based on different companies and their consents or different research protocols, a patient can decide whether or not, they want to hear that knowledge. That becomes especially important for those of us in pediatrics, because oftentimes we're sequencing somebody who isn't old enough to be able to make those decisions. So parents need to decide, do I want to know this information about my child ahead of time? Do I want to know if they carry an Alzheimer's disease risk factor gene? Or do I want to know if they carry a BRCA mutation? And those are important parts of this genetic counseling process. Then finally, we have what are called variants of unknown significance. And these are the most common things to come out of a genetic report. And the reason this comes up is because of that pathway that I showed you, where we have to figure out in a lab setting independently why genetic and if genetic changes actually cause disease. If a genetic change has been seen in multiple unrelated families, you can reproduce it in the lab, you can do it in animal models, you can target that pathway, then we can say fairly safely that it's pathogenic. If you do all of those things and the cell function doesn't change, the animal looks healthy, we would say it's benign. However, because of the amount of time and the amount of resources and the limited number of scientists, many variants, as more people have been sequenced, are going to fall into this uncertain pathway, meaning that maybe you're the only person that's been described to have that variant. There's no mouse models. Nobody's done any cellular tests. We tried to model them on computers, but we just really can't tell if there's the possibility that that genetic change is causing a problem. So is it going from surfing to sing, or is it going from surfing to surging, which is a different word, but doesn't have the implications of having something shortened altogether. So this just sort of summarizes what I just said, that the variants of unknown significance have an uncertain relationship to disease. And so since we don't know the function and there's insufficient data to confirm that the variant's associated with disease, it's hard to know whether it is related or not. And this can be really frustrating for both physicians and for families, because this is the most likely outcome. And so if you know that you have a variant of unknown significance, how does that affect your day-to-day life? Is that something that will be a source of stress and confusion because you know you have a genetic change, but we don't know the reasons for why it's there or if it's doing anything? The other issue that comes up is that a lot of phenotypes, a lot of the changes that we all have, are not just due to sequence changes. So you can get a negative report because it's not related to the DNA sequence. So as I mentioned before, we go from DNA to RNA to protein, and then multiple proteins working together cause the phenotype. But that's not the only determinant of the phenotype or the disease picture. RNA is regulated in different ways related to the amount of RNA, promoters, stability, changes in how the protein, how the genes can be accessed when they're in the chromosome. There's different cells and tissue types, different regulations in different types of immune cells. And then we have something called alternative splicing, where maybe, an RNA that's a foot long, let's use a foot as a pretend example, could be made and makes a certain type of protein, but an equally effective half-foot long RNA could be made that makes a protein that has a slightly different function. On top of that, protein expression and function is regulated. It's regulated differently in different cell types. Proteins are made in different amounts. They have different levels of stability, different levels of degradation, and they're modified in a number of different ways afterwards. Effects on any of these parts can also lead to a disease phenotype, but would not be picked up on sequencing. So then some people will say, this all just sounds really frustrating, and I know I can go on Amazon and get a $99 kit for 23andMe or for Ancestry.com or whatever. And I think it's really important to know that this is not the same type of sequencing. This is what's called qualitative genotyping. So they look for certain clinically relevant variants just across your genome. And the idea is to report potential health risks, carrier status. That's how they can help determine what background maybe you came from. Those types of sequences, while interesting and can be fun, they are not intended to diagnose any disease. So I think the most important things here are, I hope I've either reminded you of what your high school biology class was like in terms of what DNA and RNA and proteins are, but also to say that genetics have improved diagnosis and have really helped us to improve therapy and our understanding of the immune system, but it's not always as straightforward as we would like. And because of that, because of the implications to both yourself and other members of your household and your extended family, genetic counseling is incredibly important at every step of the process. Because of all of these things, follow-up testing may still be needed to determine a conclusive diagnosis. It may not just be the gene. It may not be the easy answer that we all want it to be. And then most importantly, it should not be a prerequisite to get treatment. I think, as I said from the very beginning, knowing a genetic test should not inhibit you from being able to get treated and have an improved quality of life. It shouldn't be a barrier to improving your health. So I'd like to thank everybody for their attention. I want to thank everyone at IDF for inviting me back again. I really appreciate and value these opportunities. So thank you.
Thank you so very much. What a wonderful talk. Thank you so much, Dr. Broderick. Okay, so now we are going to have you continue, please, submitting your questions about this topic. And Dr. Broderick will answer as many as possible during the time we have allotted. But before we continue, I would like to invite Eric Pluckhorn from Octapharma for a few words from our sponsor.
Hi, everybody. Hope you can hear and see me. I just wanted to say thanks to the IDF for allowing us to support these great educational forums. Thanks to Dr. Broderick for that very informative talk. A lot more complex than I'm used to, actually, but super important. And just wanted to wish everybody a happy and safe Thanksgiving. I'm sure there's a lot to be thankful for, and look forward to talking to everybody tonight with Joanna Maltese, one of our clinical nurse educators, in the breakout rooms afterwards. Thanks.
Thanks so much, Eric. So right now we are going to start having Dr. Broderick answer all those questions that you submitted. First question is, I will be going to the PI clinic at Mayo in 2 weeks. Both of my children seem to have the very same manifestations as I do. I have CVID. What should I ask the doctors there to test me for so that I can then identify the genes making me sick and maybe know, have a better idea of what to check my daughters for?
So that's a great question. I think if we think back to the CVID I put up, we know that there is a small percentage of people that carry inherited CVID, so only 2 to 10% of people. And that can be the result of a whole host of genetic changes. So I think first and foremost, when you see your immunologist talking about, is this CVID? You know, is this my diagnosis? What sorts of tests do we need to do to confirm it? What do we need to confirm the diagnosis and get treatment? But also just asking, would genetic testing be appropriate since we have the same symptoms? Because they should be able to tell you if they think it's going to be beneficial or not. You know, one of the things that we always do as physicians is, as soon as we hear a story, we start putting together all of these different pieces in our mind and trying to work down different pathways for what a diagnosis is. And I think one of the important things to remember is that, especially with immunodeficiencies, because of their rarity, you want to see a specialist because people get misdiagnosed all the time. And one of the things we know most about CVID is that it's more common than we realize, but it's also variable. And so we want to be sure that you're being treated and tested for the right things. And a lot of those things can be functional studies before you even get to genetics. And so working with that physician to see, what do I need to get my diagnosis and to help my daughters, and what can we do as a family, and present it as a family approach, I think will give you the most information.
How do you get testing done when your immunologist says she's unfamiliar with genetic testing, and the genetics department at the local hospital wants you to have other medical tests done first, neurological testing, for example, and then said they will pick and choose what genetic tests they should do? I already have CVID diagnosed, and 2 generations in my family already have it.
So one of the big challenges that we hear all of the time is because even though the field of genetics has exploded, the number of actual geneticists is actually quite small. And so for a lot of centers, the geneticists become kind of the gatekeepers to try to figure out who are we going to be able to get sequencing for, because We want to make sure that we're telling them the right information. I think that one of the greatest disservices that you can do to somebody is telling them they have a genetic variant that hasn't been validated and isn't actually true, because you've sent them down a completely different diagnostic path, because everybody is like the genes, the sequencing, are the be all and end all. And so what they're trying to do with that functional testing is to see, doesn't matter what the sequence is, what's the end result? What is that end result, and is that something that we can target? Because that end result is the culmination of the genetic sequence, the translation into the proteins working together, and all of those things. So those are very important tests. I think the other important thing is that if you have a physician who says, I don't know, which I think is the greatest thing a physician can say, and I don't think it's said often enough. Quite frankly, I think that if you don't know, we should all be honest with each other that we don't know. But I think the next statement should be, is there somebody that you could refer me to who may know the answer or who may have a suggestion? And that way you can get all of your questions answered.
I have two brothers. One of them, like me, has been diagnosed with CVID. The other one does not have CVID. We've always wondered why we have it and our brother does not, but we've always wondered what the likelihood will be that any of our children might end up with CVID.
So one of the challenges with CVID, because of the number of different genes that have been identified and the very few number of families that have been found where it's a clear genetic diagnosis, is that there's a concern of something called modifier genes, meaning that it's not just a one-to-one, you inherited this gene and that's why you have CVID. In some families, it seems like you need a couple of genetic changes to come together. Those are very difficult to prove in the lab. So the only way to be able to predict, if your children were likely to have an inherited version is if you fell into one of those small families where you can say with a known variant, yes, this has been seen in multiple other families. We have this exact same genetic change. Otherwise, it would be diagnosed the same way in your family as it is in other families with CVID by looking for autoimmunity or recurrent infections and having the lab work done. CVID is one of the biggest genetic challenges.
My genetic testing from Invitae showed heterozygous variants for DOC8 and ITGB2, but my immunologist is not sure how to interpret this, especially for the ITGB2 variant. In both cases, there is sequence change. Is it worth pursuing further testing?
So those genetic changes fall sort of in that VUS category, most likely. And so those are the biggest challenges for all of us because, for what we know today, does that genetic change actually mean anything? Is it changing the word name, going back to my surfing example, how much is it changing it? And it becomes a VUS a lot of times when we can't predict what it's doing. And so there's two approaches that I usually recommend. One, as I keep saying, we're learning things every day. So those genes are not going to change, but maybe somebody else 5 years from now will have those exact same genetic changes. And you can just keep following up with your immunologist to say, hey, has anybody else, or your geneticist or your genetic counselor saying, has anybody else been identified that has my disease phenotype and has these same genetic changes? The other things, that I think are more important is to talk with your doctor in terms of if you think your treatment is working. If you think your treatment isn't working, then you can talk about pursuing variants of unknown significance. But as more people are sequenced, we know we have those 4 million changes that make us us. And so not all of them are going to be the cause of disease. Some of them are just the things that, make our hair straighter or make our hair more curly and things like that. And so you have to weigh with your physician the risks and benefits of other testing based on how your current clinical picture is. It's not like when you get an immunoglobulin level and it's an absolute number. Genetics right now is a moving target because we're learning new things about all of these genes every day.
I have CVID and rheumatoid arthritis. have genes been identified that show that somebody can have both of these? While I understand it is common, I've read that up to 30% of the people have this. Is there any information that you can share?
So people with CVID can have autoimmune manifestations, and a lot of times those are even more common than the infectious complications. The way autoimmune disease has been approached has been a little bit different. They tend to do big studies on populations because autoimmune diseases are so common. And so they have certain risk factors that they say, you might have this genetic change that makes you more likely to have an autoimmune disease. Those are called GWAS studies. And those sorts of studies are constantly being revised to see, is there something that could be targeted for therapy? But in terms of a one-to-one genetic link between rheumatoid arthritis and CVID, there's not one that I've read about.
I've had genetic testing done, and it came back as monoallelic mutation of TNFRSF13B gene. Is there any specific thing I need to do? My immunologist just told me it's confirmed and that was it. Do you recommend anything else?
So that's one of the genes that is looked at in terms of being a modifier gene. And so one of the things that comes up is that when we're trying to figure out how a gene may lead to a disease, we have to consider, what do we know about the human population as a whole? How many times has that gene been seen, and how many times has that genetic change been seen? And so one of the things that becomes complicated in the gene like that, which makes a protein called TACI, which is much easier for me to say than TNFRSF13B, is that depending on the variant, you can find those genetic changes in 10 to 15% of the population who are otherwise healthy. And so it gives you some information that maybe you have a modifier gene, but that is one of the biggest areas of study right now in CVID, is to see, are there, just like the picture I showed you for CGD, are there certain genetic changes within that huge gene that could give us more predictive factors for how severe your CVID will be, if you would respond better to different therapy and things like that. So for right now, what's done for patients with those variants is that we still, treat CVID the same way. We want to make sure you're not having the infectious complications. We want to make sure you're not having autoimmune complications, and we want to be sure that you're on the right levels of IVIG if that's what you need, or sub-Q IG.
Is genetic testing done via a blood draw?
So it can be done by a blood draw or by what's called a buccal swab, which is where you take a Q-tip and put it on the inside of your cheek, and that just scrapes some of the cells off. Or some companies do it with a spit kit, where to sort of go back to my childhood, you huck a loogie into a little container and send it back to the company. So there's different ways of doing it.
I have an IgG3 deficiency and no one else in my family does. Does that mean that it was caused by a recessive gene in my parents? Or are there any types of primary immunodeficiencies caused by environmental factors?
So those are good questions. So one of the things that we didn't talk about, but if I'm invited back in the future, I'm going to bring this into my presentation, is that when you're making antibody, it is a long gene, and you actually take pieces out of that gene and splice them together, and then mutations happen on purpose, and that's how we're able to make antibodies to all sorts of different things. When those splicing and those, like the cutting and pasting together, is happening, you have to be able to glue it on to what's called the heavy chain. And that's how we get all of the different types. You get G, A, M, E from whatever one is kind of glued onto the end. Those are all different genetic components. So there's two possibilities. There's the possibility that your levels are quite low and below the limit of detection of the test. there's the possibility that you're missing a section of that gene and it can't be glued onto the end. There's the possibility that your cells are just opting, because this is all by chance, to not glue it onto the end. I have not read anywhere about a recessive inheritance of an IgG3 deficiency, though. I think there was a part of the question that I missed at the end. Was there something, Kathy?
Is primary immune deficiency,
Oh, environmental factors, yeah. That has not been described. There are certain things in the environment that may affect how your disease is perceived. One of the things that's an active area of study right now is what is the impact of your own gut microbiome on immunodeficiency, and is that related to barrier dysfunction? Is it because cells are inappropriately seeing the normal bacteria and so they're attacking it? That's an active area of research, and that would be the closest thing to a relationship between the immune system and the environment. But being exposed, outside of Chernobyl, and having complete erasure of your bone marrow in a nuclear explosion, there isn't a specific environmental factor that we would commonly see in the US that would cause a primary immune deficiency.
We have several questions from people saying, my doctor just will not approve genetic testing. Is there anything I can do?
So I think, for a lot of people, the challenge is, if you get this test back and you don't know what it means because it has all of these variants, are you actually helping somebody at the end of the day? And for us, that's our biggest concern, is we want to make sure we're ordering the right tests to help you. And so for a lot of people, that's why they then ask for a referral to a genetic counselor to talk about the different options and talk about whether the genetic tests would make sense for them and their family. And a lot of this just comes because all of this information is so new. For most physicians, once you get past that DNA, RNA to protein, that's the end of the genetic education in medical school, for a lot of physicians, because genetic testing by Illumina, the idea of exome sequencing, all of that stuff was just in a research world up until a couple of years ago. And the fact that it's been approved to go into the clinical scenario just left a lot of people without that knowledge. If you haven't been in the lab and looked at the sequence yourself and done that, which is hours of, your own time and trying to learn those things, not everybody has those opportunities. And so they're doing it in a way to make sure that you're not misinformed and to provide you with the best care. And so I would go the next step then, if you're interested, I think talking to a genetic counselor is really the most important thing that you can do, because there are so many limitations and challenges related to the testing.
If someone has very low immunoglobulins but does not experience abnormal infections, could that be a sign that the genes are somatic, as you described? If so, are there any implications to consider with genetic testing?
So there's, we know because of all of the patients that have common variable immunodeficiency that it can take years to develop. And so it doesn't necessarily mean that it's a somatic variant. It may mean that it's a combination of genes that have to come together and a combination of changes that have to happen over the course of someone's life. Because as I talked about with the variable expressivity, we know, for example, with TACI, we know that when you're a baby, the TACI protein expressed by your cells is very low and it increases with time. And so that's a theory with, if you don't have experienced the normal increase in protein, could you have symptoms? So it doesn't necessarily mean that it's a somatic variant. It may just be, it's not expressed at the right time or in the right amounts or things like that. So I think it could be a very challenging question to ask for with respect to genetics. And so again, going back to this idea that the core, for immunology is still doing a lot of functional tests to say, what is the end product of the genetics? Are we making immunoglobulin? What are we seeing? The most important thing that we can see as physicians is what does our patient look like to us, either by Zoom or in the clinic, and how are they doing? That's the bottom line.
Thank you so much for answering all these questions. We are at the end of the Q&A time, and we have lots more questions. So Dr. Broderick, we might send a few your way if that's okay.
Okay.
And then follow up with everyone whose question wasn't answered. So first of all, again, thank you. Thank you so much, Dr. Broderick, for taking time out of your busy day to volunteer your time and be here with us. It's because of amazing volunteers like you that we're able to provide these forums. And to everybody here who submitted a question, thank you. Thank you so much. And even if you didn't submit a question, thank you so much for being here. Now I would like to invite Sean McCabe to say a few words from our sponsor.
Thank you, Kathy. Can you hear me? I think I hit the right button. Yep, good. So I'll echo the thanks from the peer set to the IDF, the staff, Hector acknowledging all the great effort it takes to stand these up, and certainly standing up so many of these since the the impact of COVID has been phenomenal. And so in the spirit of Thanksgiving, just a sincere thanks to staying connected, educating the community, and most importantly, empowering them. 111 participants I still see on, which is just phenomenal. And seeing these week in, week out has been just amazing. So thank you for that. Dr. Broderick, all I can say is wow. Unbelievably insightful content, very sophisticated delivery. I love the one thing I could connect with was Hakalugi from a childhood perspective as well. But certainly stayed as much as I could in terms of the content, really well delivered and incredibly insightful. So thank you. And just a very brief introduction. I have the honor and the privilege of leading the Immunodeficiency franchise at Takeda. If you don't recognize the name, our lineage starts with Baxter. We're the proud manufacturers of Gammagard, HyQvia, and Cuvitru. And I've said before, hey, the name of the company may change, but our commitment to serving the community and most importantly, delivering safe and efficacious therapies certainly remains. But we're not attending these forums to talk about our product. We're here to most importantly listen. We only know what we're told in terms of what your needs may be, and we certainly want to address any questions you may have. And to that end, I have my colleague Dana Flathammer, who leads our IG community support team and my IG source to be able to give a brief overview of the products and services that we offer in that environment. And so from that perspective, again, just my sincere thanks and look forward to seeing you in the forum hereafter.
Thank you, Sean. Now let's get ready to move into the virtual exhibitor hall. And before we do that though, just a reminder to everybody to please take a look at the IDF website, take a look at our publications, our calendar of events, anything and everything we offer is available there. And you can also follow us on social media to keep up to date with our community. So please check it out. Once again, thank you to our sponsors for making this forum possible. Thank you so much for being here tonight, and take care. Have a nice Thanksgiving. Be safe. Take care. Bye.
Thank you.